So that concludes our speakers for the evening. And so what I wanted to do is just take a couple of minutes really to hopefully summarize a little bit about what you've heard. So the centre opened just over 5 years ago in April, 5 years ago, and since then I think we've come a long way. We have over 100 people, scientists, basically working in the building, and just to recognise that the building itself was funded entirely by philanthropy. So the building wouldn't be here without the generous gifts of donors. And really what we've tried to do in those 5 years is, build an infrastructure, some of the things you've heard about from Tim and James, where people can come and work on cancer immunology in an environment that allows them to really work at the cutting edge. We provide some of the best facilities. We have very good scientists. We have lots of good infrastructure around us, which allows us to really ask the difficult questions in cancer immunology. None of that happens alone. As we mentioned, we're just across the road from the hospital. And so we work very closely with the NHS, our clinical colleagues. And one of the key things, I think, over the last 5 years has really been the recognition that actually working on primary human samples, which patients donate, that are removed from surgeries and people give permission for us to work on. And that's absolutely crucial for us to understand really what cancer is and how we can go about making better treatments for it. And so thank you to the patients that donate their samples and also to the interaction that we have with the NHS. And then Charlotte mentioned it, the clinical trials unit here in the centre. It's a fantastic clinical trials unit. They're developing clinical trials, both the kind that Tim mentioned, which is what we call a phase 1 trial, where we take something that we've observed in the lab and try and test it for the first time in patients, in humans. And then also what we call phase 3 trials, which is where we're trying to see if our new immunotherapies are as good or better than the current gold standard for treatment. And sadly, in some cases, to be better than the current best treatment is not very good. And Charlotte alluded to it, the survival rate at the time she went onto her trial was 20% of people in 2 years. So, and Tim mentioned it as well, which is we want to move that curve basically higher and higher with an increasing knowledge of what cancer immunology and cancer immunotherapy treatments can do. And so really that's what the centre is. And thank you to those of you that have donated and been interested in the centre to date. And I think now we'll move to our Q&A session. I think there's been a series of questions that have been put in the chat. So I will take them essentially from the top down. And I will ask James to take all the difficult questions, Tim to take the second most difficult questions, and I will try and take the easy ones. Charlotte, feel free to take any ones that you'd like to. Okay, so the first question is, do some cancers first successfully treated by immunotherapy evolve to evade the immunotherapy treatment to then potentially reoccur in this evolved form? So that's a really great question. And sadly, the answer is yes. What we see when we use lots of different types of immunotherapy is that we might see an initial good response that the tumor might shrink. But in some patients that then, after a period of time of receiving the immunotherapy, the tumor returns. And I'm sure all of you have heard of Charles Darwin and the theory of evolution. Essentially what that says, if you apply a selective pressure to an organism, it will basically, there'll be an evolution that it tries to basically survive that selective pressure. And the same is true of cancers on an individual single cell level. Every individual cancer cell is trying to survive. And so in some cases, we have effectively what becomes a race between the immune system and the cancer cell. The immune system is capable of recognizing some of that evolution that the cancer can do. But at the same time, the cancer is able to suppress the immune system. So it's really whether our intervention can sufficiently augment the immune system to allow it to eradicate the tumor before it can evolve away new defense mechanisms of the immunotherapy is really what the kind of race that we're in that kind of context. Tim, James, I don't know if you wanted to add anything else to that one.
Just that it's one of our major research interests in my lab is how that process of evolution happens. And we study some proteins that exist in cancer cells that are normally there to mutate viruses to stop them infecting us and to stop them being able to replicate. And they can actually mutate our own DNA in cancer. And our hope is that by understanding these enzymes, we may be able to block their activity and therefore limit the cancer cells' potential to evolve through gaining more mutations during treatment. So it's definitely a really important area and a big interest to us, certainly. Yeah.
James, did you want to add anything?
Just a small detail about how the immune system can target different targets within cancer, as I talked about. And some of those are much harder to mutate than others. So if we can discover which ones are common to cancers and cancers tend to keep them over time, then that can help us develop more robust immune responses.
Great, thank you. So if we move on to the second question, and that is, what impact does immunotherapy have on those with autoimmune diseases? So again, really, really great question. And I think at this point, it's probably useful to kind of split apart the idea that immunotherapy is one thing. Conceptually, it is one thing. It's using the immune system to fight cancer in this context. But actually, there's lots of different ways that we can do that. And they work in slightly different ways. So the antibody that Tim spoke about, the CD20 antibody, works by deleting the tumor cell. So we have a cell in the body called a macrophage, which is able to actually physically engulf and destroy tumor cells. Whereas the CD27 antibody Tim mentioned is what we call an immune modulator, an immune stimulator, which stimulates T cells and another type of cell called a natural killer cell. And so you can probably imagine that those two antibodies might work in different ways. And because of that, they stimulate the body and the immune system in different ways. And so they might have different impacts on the body in general. And so immunotherapy in the context of people that might have autoimmunity, it's very much a case of what immunotherapy and what type of immunotherapy. So for instance, actually, the rituximab antibody that targets CD20, that targets B cells, is actually used for the treatment of certain types of autoimmunity. So it's used in rheumatoid arthritis and certain other autoimmune conditions. So in that context, it's very beneficial for someone with autoimmunity. It's actually used as the treatment, whereas you might not necessarily go ahead and try to stimulate the immune system with a T cell agonist or an immune modulator like CD27 that Tim mentioned. Similarly, with the checkpoint blocking antibodies, again, whether you would necessarily go in that context where there might be an ongoing immune response, such as in autoimmunity, yeah, you probably wouldn't use those in that context. But it really comes down to the type of autoimmunity, whether it's B cell mediated, T cell mediated, or otherwise, and the type of immunotherapy that we're talking about. Tim, did you want to add anything to that one?
No, I think they covered it very well, Mark.
James or Charlotte, any additional context for those ones? Okay,
Not
great.
for me.
No, that's great. The next question is, how do we join immunotherapy trials? So really good question. I mean, maybe Charlotte, if I throw that one to you first, how did you join your immunotherapy trial?
Yeah, I was invited to apply. So there was this clinical trial running, and I kind of fitted the overall cohort of people that they were looking to recruit for the clinical trial, and it was specifically for people with stage 3 melanoma to try ipilimumab. But it was brought up to me, was mentioned by my surgeon at the time and the oncologist who kind of were treating me with more traditional treatments at the time at Southampton. And so I spoke to them about it and got their view and opinion, and took away quite a large amount of material actually to understand what the impact would be, what it would mean to me, and how it would impact my life, and whether I really wanted to go on that journey. So that's how I got involved and got invited to join the trial.
Yeah, thank you. And I think that's pretty common. I think normally it's at the recommendation of your clinician. That should be your first port of call, really. They'll know from you the specifics of your particular tumor and the stage and everything else, whether you're suitable for a trial that might be running nationally or even locally. So I think that should always be your first port of call. Okay, next question. Why does immunotherapy only work on some types of cancers? So that's a great question. And one of the things that I think all of us that are working in cancer immunology are interested by. One of the determinants Tim mentioned in his talk is whether you have T-cell infiltration, whether you have lots of T cells in there is one of the things that could determine whether you might respond to a certain type of immunotherapy. Another thing is how many mutations you have, which again relates to things that both Tim and James kind of are working on. So mutations are basically the thing that makes the difference between our host cells, our normal cells, healthy cells, and the tumor. And each individual tumor from an individual will be different from another. And actually within an individual's tumor, the genetic mutations will be different between each tumor cell in that individual's tumor. There's a huge amount of complexity, but certainly the correlation with immunotherapies has been the more mutations you have in your tumors, the easier it is for the immune system to detect and potentially destroy it. There's a good correlation with the checkpoint blockers, anti-PD-1 and ipilimumab and antibodies like that. That there's a good correlation there between genetic mutation and response, also with whether or not you have T cells infiltrating your tumor or not. So they're just a couple of the things. In terms of the tumors themselves, although each individual tumor is different, certain types of tumors tend to be more or less infiltrated with T cells. So that's one of the things that can determine it. There are also certain tumors that have particular mechanisms of immune suppression that are very common. One of the things that Tim mentioned that Gareth Thomas is very interested in is a cell called a fibroblast. That's a cell that actually can wall off a tumor and stop the T cells getting in. So if the particular tumor type has lots of fibroblasts, you can see how that maybe then the T cell therapy might not work so well. Tim, do you want to take over from that one?
Sure. And I think that the things that Mark has mentioned are state of knowledge at the moment, and it's an area in which we really have a long way to go, I think, understanding which tumor. So although there are some general rules of thumb emerging, if you like, so generally a tumor with more mutations will respond better to immunotherapy, and tumors that are full of T cells are likely to respond better. It's not the case that an oncologist could look at someone's tumor and say, you're definitely going to gain benefit from the therapy I'm about to give you on that basis. We just don't understand enough of how that all works yet. So that's a really intensive area of investigation at the moment, and will continue to be so over the coming years, to try and work out which tumor is going to. Obviously, you don't want to while you want to spare someone a therapy that's not going to work for them, on the flip side, you certainly don't want to withhold a potentially useful therapy from someone just because they have maybe a low tumor mutation burden or not much PD-L1 expression on their tumor. So getting these biomarkers right is really, really important to understand. So that's a major focus of research.
Yeah, thanks, Tim. So maybe we'll move on to the next question, which is, why do some people not tolerate immunotherapy? And I think that really kind of relates to the point I was trying to make about the autoimmunity. Every individual is different. So the way that your body responds to immunotherapy, to immune stimulation, is different. Sadly, we've all seen that through COVID. Different individuals respond differently to the vaccination. People were given the same vaccine, and yet their responses to it were very different. So both in a good way, in terms of some people generated very good COVID responses, but also some people had detrimental responses or didn't respond very well. That's all down to the individual genetics of every person. So there's a number of things that can play a role in whether or not someone can tolerate or not tolerate a particular type of immunotherapy. And again, it might depend on the type of immunotherapy. You might be able to tolerate, a rituximab type antibody, but not so much an ipilimumab, or you might be able to receive CAR T cells, which we haven't really spoken about much this evening, but that's another type of immunotherapy. But again, another patient might not be able to tolerate that. Generally, the fitter and healthier you are, then the better you're able to tolerate cancer treatments in general. And to a certain extent, that's true for immunotherapies. Did anyone else have anything to add to that one? Great, so we'll move on to the next question, which is, do you think immunotherapy treatments can be developed for all cancers, or does it depend on where in the body it is? So I'm by nature an optimist, so my answer to that is yes. And just simply from a point that Tim made, which is really, we're at the start of our understanding about how cancer and the immune system kind of co-evolves and how it coexists. And James mentioned that when you actually see a tumor develop, it's already evaded the immune system, right? So all the time your immune system is finding and deleting tumor cells or potential tumor cells. And so I think the idea that if we understood that process better, and if we understood how the tumor cells and their tumor microenvironments suppress the immune system, then we can make therapies that allow us to overcome that. And checkpoint blockers and other therapies are our current best ways of doing that. But it's clear from increasingly sophisticated studies that we're doing now that this evolution of the tumor under selection from the immune system is finding ways of trying to evade it. And so it's clear that we need to understand things in a much more complicated way than we used to. At the start of my career, we used to look at a single receptor and a single signaling pathway. We're now in what we call an omics era, where we're trying to understand the signaling pathways that are happening in the cell all at the same time. So we have 20,000 genes, and each of those can be on or off. It could be at a low, medium, or high level. So if you think about that as a system that we need to understand, it's incredibly complicated. But I think with enough effort and time, then I think we can understand it. It's not ultimately, it's not a magic box. It's a system that responds to stimulus and suppression. So I think we can find ways eventually of being able to treat these different types of cancers. The question about where in the body it is, and I see there's a question a little bit lower, which is about crossing the blood-brain barrier for brain cancers. So that's a really interesting question, and that's so generally immune cells don't cross the blood-brain barrier, the T cells, very, very frequently. But actually people are designing antibodies and other therapies that actually can cross the blood-brain barrier. We use a receptor that's on one side of the blood-brain barrier and use that as a shuttle to take antibodies across the blood-brain barrier. So people are designing those things. So really clever scientists in industry and in other academic institutions are using what they call things like the brain shuttle, which allows things to cross that blood-brain barrier. So I think we will be able to treat cancers that are in more what we call immune privileged sites. I see I'm getting a prompt to say this is the last question. What is the main challenge in immunotherapy research? So that's unfortunate. So that's going to take me at least 45 minutes to answer that one. But I would say the complexity. That would be my response. It's incredibly complex. It's incredibly dynamic. It responds and it changes. That would be my response. Tim?
Yeah, there's so much to understand still, and so many targets that we haven't even discovered yet So yeah, there's a lot to be done.
Yeah, James, as a newbie at the start of your career.
Definitely the complexity again. Cancer, it's not one disease, it's hundreds, thousands, all with different best ways of treating it. And trying to find different ways of tackling this, a sort of toolbox that we could use in clinics, is quite a big challenge to tackle.
Great. Charlotte, did you have anything to add?
No, I didn't have anything to add other than I'm just quite in awe of the work that you guys do. I think it's very impressive, and it's been I'm probably going to say something, but it's just been really informative, and to get an update on where the immunotherapy treatments have been going. Certainly since I started on ipilimumab, that was the only one, and it sounds like there's many available now.
Great. Thank you very much. So thank you everyone for joining us tonight. So that's the end of our Q&A. As I mentioned, we'll try and respond to the questions that are left in the chat. So you've heard from some amazing people. Hopefully you agree, and that we're all coming together to hopefully try to find ways of fighting cancer in a better way. So obviously James and Tim are on the front line of that, developing new ways to diagnose and treat cancer. And obviously we wouldn't be able to do that without people like Charlotte taking part in clinical trials, because that's ultimately how we test what we're trying to develop in the lab. And obviously that happens with a fantastic clinical trials unit and partnership with the NHS, as I mentioned. So the other person that's obviously important in cancer research is you guys. As I mentioned, nothing would have happened in this centre. We wouldn't have the centre without philanthropy. So that's through generous gifts of you. And hopefully some of you will consider making a donation to the centre tonight. You can make a donation by post, by phone, or by visiting www.southampton.ac.uk/donate. The details should be on the screen, and we'll circulate them by email after the event has closed. And in a minute, you'll see a short poll, which will come up on your screen, which we'd like to ask you to complete. So that's really just, we'll take a couple of minutes of your time and gives us some feedback on whether what you've heard tonight is useful, informative, if there are ideas you have about other things you'd like to hear about. And for any of you that would like to watch what we presented tonight again, we'll be sharing the footage over the coming days. So keep an eye on your inbox for that. And you'll also find details of how to join our mailing list, so we can keep you updated on future lectures. So I've gone 1 minute over. I'm very sorry, but thank you very much for joining us, and good night.